Welcome to the Adelaide Biomed City Minireview webinar series. My name is Andrew Zannettino and I'll be the chair for this session this afternoon. Before I introduce our first speaker, I'd like to acknowledge the Kaurna people, the original custodians of the land on which we meet here today. And I'd like to pay my respects to their elders, past, present and emerging. So I'm delighted to actually introduce our two speakers this afternoon, Dr. Danny Wilson and Professor Michael Beard. Our first speaker this afternoon is Professor Michael Beard. Michael heads the Viral Pathogenesis Research Laboratory at the University of Adelaide, and that investigates the host cellular responses to the viral infections with a focus on Flaviviridae family members such as hepatitis C virus and the arboviruses dengue, Zika and West Nile viruses. Michael uses cutting-edge molecular methods such as CRISPR genome-wide screens, liver organoid cultures and mouse models of viral infection to characterise the host responses to infection. Michael's work has been continually funded by the National Health and Medical Research Council for the past 20 years, and he is currently Deputy Director of the Research Centre for Infectious Diseases and the Head of Department for the Molecular and Biomedical Sciences in the Faculty of Sciences. It gives me great pleasure to welcome him today to present his presentation entitled Immunological and Cellular Control of RNA Virus Replication and Pathogenesis. Please welcome Michael.
Thanks, Andrew, for that introduction. Just let me share my screen here. As Andrew said, we're part of the, or Danny and myself, and Danny will be giving the second seminar today. We're part of the Research Centre for Infectious Diseases, and we're located down on the corner of Frome Road and Victoria Drive, probably just about as far as you can get from the Biomed City. But we do have lots of collaborations with people within the Biomed City, and we're always open for new collaborations. So if you want to talk viruses or parasites, we're the people to talk to. So the Viral Pathogenesis Research Laboratory, we're really quite diverse, but we really have an underlying theme in that we're interested in investigating the host response to virus infection. And so this is the really early response, or the early innate response that happens within hours of a virus infection, namely the interferons and interferon-stimulated genes. And so we have programs around characterising interferon-stimulated gene control, identification of novel interferons, and we use sort of cutting-edge technologies such as genome-wide CRISPR screens and activator screens to answer those questions. And I'm going to talk a little bit about some of those screens as we move through the talk. Mostly towards the end. We've also have a group looking at developing new model systems to study virus host interactions. And through some interactions with the Royal Adelaide Hospital Gastroenterology Department, we've been developing human liver organoids. And this is just a picture here of one of those liver organoids that's infected with hepatitis B, shown here in red. So we're trying to develop models that are the closest as we can get to what's happening in vivo. And then we have projects looking at innate immunity to hypoxia, but I'm not going to talk much about that today. I thought I'd just introduce the flaviviruses to you. You probably don't think about these viruses very much, but some of them that you may have heard of include dengue, Zika, West Nile virus, we're going across there, West Nile and yellow fever virus. And these viruses are endemic to much of the world's population, certainly in some of them are in northern Australia, and they really do cause significant morbidity and mortality. There's no good therapies for these viruses except for a vaccine for yellow fever virus. So they really are a major medical problem the world over. I'd also like to introduce the interferon or the innate immune system to you as far as at the cellular level in response to a viral infection, because it underpins a lot of what we're going to talk about in the next 8 or so minutes. Essentially, when a virus infects a cell, and we're just using the Zika virus here as an example, and we're not going to go through this in great detail because of time, but essentially it sets off a whole number of canonical pathways shown here that result in the activation of transcription factors and the production of the interferons. And these are the type 1 interferons. And this is why you would feel so sick following the flu or perhaps the common cold infection. It's these interferons that elicit that feeling of lethargy and muscle ache, muscle weakness, etc. So these type 1 interferons can act on the cell that they're being produced by, but they can also act on the neighbouring cell. And sorry, I haven't quite got this right. They can also act on the neighbouring cell to activate what we call the JAK-STAT pathway. And it's this JAK-STAT pathway that's so crucial to controlling viral infection in the cell. And what these interferons do is they activate 300 to 600 genes, depending on the cell type. And most of these genes are either antiviral, or they're involved in inflammation, or they're involved in regulating the whole inflammatory response. But the end game here is really to set up an antiviral state. Now, sometimes the virus wins throughout this process, and that's when you get disease or a viral infection. Sometimes the cell wins and the cell can clear the virus from the cell. But we're interested in looking at new novel interferons, and we do this with our collaborator, Paul Herzog at the Hudson Institute for Medical Research. And Paul discovered this novel interferon called interferon Epsilon back in 2013. And so we're really interested in how this interferon acts in the female reproductive tract, because it's constitutively expressed in the female reproductive tract. Now, that's different from most of the other interferons that I've talked about before, because you may remember that they're actually induced following a virus infection. So this is actually constitutively expressed. And you can see here very nicely that this interferon epsilon is expressed within the female reproductive tract. There's an immunohistochemistry staining, and also some real-time PCR compared to the other interferons. So we wanted to ask, well, what is the antiviral role of interferon epsilon in Zika infection of the female reproductive tract? Because Zika virus is now a recognised sexually transmitted virus, as the virus actually hangs around in the seminal fluid for anywhere between 3 to 6 months following infection of males. So it's a significant problem, because that virus can then, if the female is pregnant, can actually cross the placenta and infect the fetus. And we all probably know about the devastating effects of Zika virus infecting the unborn child. So we started a program to see whether, in fact, these cells of the female reproductive tract could, in fact, be infected with Zika virus. And yes, they can. Cells shown here in red. VK1 cells and vaginal cells, VK2, actually can be infected. And if you treat these cells prior to infection, you can see here that this interferon epsilon, if you treat with interferon epsilon, you can see that the virus is significantly reduced, both at the RNA level and the infectious virus level. So clearly, interferon epsilon has antiviral properties. We're very fortunate to have an interferon epsilon knockout mice that was supplied by Paul. And if you infect these mice, or if you use these mice, you infect them with Zika virus intravaginally, you can actually see that mice that don't have any interferon epsilon have significantly more virus compared to the wild type, certainly at day 5 and day 7, and possibly a little bit at day 2. So clearly, interferon epsilon is playing a key role in the female reproductive tract to protect these mice from Zika virus infection. We did some studies where we then added to wild type mice, we then added antibody to interferon epsilon, and that's a neutralizing antibody. And when we do that and then challenge these mice with Zika virus, you can see that in the vaginal washes of these mice, there's increased virus replication. So we've knocked out a complementary study to our interferon knockout mice. And then conversely, if you add recombinant interferon epsilon to these mice intravaginally, you see a decrease in virus replication. So this is strong evidence that interferon epsilon actually can play a role in the female reproductive tract. I'll put this slide up just to give you an idea of the sorts of genes, or the number of genes that are activated in response to these interferons. There are hundreds of them. This is just a handful of them. And you can see that they segregate out quite nicely based on whether it's interferon alpha, interferon lambda, or interferon epsilon. But what the really interesting thing from this work is, and you can't really tell from this study, was that interferon epsilon produces more of an antiviral phenotype compared to interferon alpha, which produces a more pro-inflammatory response. And if you think about it, this is really what we would want to have in the female reproductive tract. We don't want to have expression of an interferon that is driving a strong inflammatory response. We'd rather have a more of an antiviral or possibly an antibacterial response. So just to summarize this part of the work, and I think we're on a paradigm shift here, and because interferon epsilon is constitutively expressed, this is the only interferon that is expressed that way, and I'll explain it to you here in this diagram. So interferon alpha, or type 1 interferon, acts through the JAK-STAT pathway, and it's produced in response to viral infection, as I showed in one of my early slides. And so that can then, the cell is then still able to infect, sorry, the virus is still able to infect the cell, and there's a bit of an arms race between the virus and the cell as to who wins out first. And sometimes the virus wins, as I've mentioned, and you get an infection. Sometimes the cell wins and you're able to clear the virus. But the constitutive expression of interferon epsilon allows the cells of the female reproductive tract to have this basal antiviral state. And that's what allows protection in that setting. So that's what we think is happening. And we're doing a whole lot of work to actually determine if this is a good topical microbicide to prevent sexually transmitted diseases such as bacterial infection and other viral infections. I'm now just going to shift gears a little bit for the last couple of minutes and just to identify some work that we're doing using CRISPR screens to identify novel host factors that are critical for virus replication. And the example that I'm giving here is a Zika virus infection. So what we're doing is here we're using, most of you would probably know about CRISPR technology, but now we can in fact knock out every human gene in the genome, and we can then screen for genes that are knocked out that will allow cells to survive in response to a cytopathic virus. So this works because these viruses the flaviviruses, are actually all cytopathic. So cells that survive following addition of a lentivirus pool of all these S-guide RNAs will allow us to look to see what guides are enriched, and then we can determine what host factors are important for replication. We've done this for Zika virus, sorry about that, and we've identified RAC1 as a candidate. And Byron Shu, a very talented postdoc in my lab, has in fact shown that if you then continue on with these sort of work and you knock out RAC1 individually in cells, infect them with different viruses that are transmitted by mosquitoes or by the ticks, you actually show a significant decrease in virus replication. You have to remember these are log scale. So there's over a 90% decrease in replication, and this is quite significant. We've also done some work with SARS-CoV-2 with our collaborators at the NIH Rocky Mountain Laboratories, and we've also shown that SARS-CoV-2 is responsive to RAC1. So the question is, can we actually modify RAC1 or learn more about it and actually develop pan-antiviral therapeutics? How's RAC1 working? Just really very quickly, just showing some of the sort of technologies that we use in our lab. This is normal cells shown here for Zika virus and for dengue virus, and you can see here that this is where the virus replicates, inside these very small convoluted vesicle packets. So the virus rearranges membranes. When you remove RAC1 through an siRNA, you start to see the real changing in these vesicle packets. And we think that the RAC1 is really important through an interaction with a viral protein for the generation of these replication factories. Just to finish up, I'd just like to mention that we're also doing activator screens, and this is Emily Kirby in the lab, again, another PhD student. And this is a modification of the current CRISPR technology where we have a dead Cas9. So essentially this Cas9 doesn't cleave the DNA, it just directs a whole lot of transcriptional activators to a site upstream of a gene, and we get transcription of the messenger RNA. And we can do this also in a screening process. So we can activate just about every gene in the human genome. And then again, like we've done before, we can then select and look for guide RNAs that are overrepresented. We've done these sorts of screens, and we've come up with a whole bunch of candidates that look like that they could be involved in protecting cells from viral replication. So now the challenge will be to actually work out what exactly all of these are doing, and that's part of Emily's PhD. So I'd just like to finish there and thank you all for listening, and just to acknowledge everyone that's been involved in this project, certainly at the University of Adelaide, and again, our collaborators both at Adelaide, interstate and overseas. And just also thank you to our funding agencies. Thank you very much.
Thanks, Michael. Terrific presentation. Michael, might start with a question if I could.
Sure.
Just in relation to RAC1, can you give me an understanding of the type of protein that RAC1 is and what its intended function is in normal cell biology? Yeah,
it's one of these proteins that it's a scaffold protein. It's also a chaperone protein. It's one of these proteins that does seem to do everything. It also has some kinase activity as well. So that adds another dimension to actually work out exactly what it's doing. It's also part of the ribosomal complex. Yeah. And some people have said that it inhibits translation, or if you knock it down, it can inhibit translation. We've found that has had no effect on the virus. It doesn't affect viral translation. So the question is, we think it acts by interacting with a viral protein, NS1. And NS1 is important. I didn't have time to go into that, but NS1 is important in setting up those replication complexes. So, do you think it's therapeutically actionable? I don't know. We haven't gone down that path. The problem will be there, of course, because it seems to have a lot of other functions, normal functions in the cell. I mean, I think time will only tell if we could actually find an inhibitor that would actually be specific for the action that we wanted. It might be one of those long bows to go down. I'm not sure at this point in time. Yeah, sure. Look, Michael, thanks so much for your presentation. Unfortunately, owing to time, I can't ask any more questions or take any more questions this time, but thank you so much for your excellent presentation. Thanks, cheers. That leads us to our next speaker for this afternoon, Danny Wilson. And Danny completed his PhD at the Walter and Eliza Hall Institute in Melbourne with a focus on vaccine and drug development targeting human malaria parasites. So Danny established his group in the Research Centre for Infectious Diseases at the University of Adelaide in 2014, where he is developing new drug and vaccine approaches to defeat malaria and developing new super resolution microscopy techniques to excuse me, to understand its complex biology. Danny is also was the recipient of the coveted South Australian Young Tall Poppy of 2019, and he's currently the Hospital Research Foundation Fellow and NHMRC Ideas Grant recipient. And the title of his presentation today is Malaria Parasite Biology and New Targets for Therapeutic Development. So please welcome Danny.
All right, thank you, Andrew, and thanks to the organisers for the invite today. Hopefully everyone can see the screen and we're ready to go. Yeah, so as Andrew mentioned, my lab does a lot of work on malaria, and malaria remains a major problem for the world today. So there's over 200 million cases of malaria every year, around about 2 million cases of severe disease, leading to around about 406,000 deaths per year. On top of that, malaria affects somewhere in the order of 9.5 million pregnancies per year, leading to impaired development in children, up to 750,000 children per year. So it has and remains a global pathogen of significance, which has long-term effects on health and prosperity of communities affected. So malaria is a eukaryotic parasite. It has around about 5,500 genes. Half of those, a large part of half of those resemble the same genes that we have. And then on top of that, around half the genes of malaria parasites have known homologs, and this is to do with their very unique life cycles. Malaria is transmitted by female anopheline mosquitoes, and of the 5 species of human malaria parasites, the most virulent and the major cause of death is Plasmodium falciparum. Plasmodium vivax is a major pathogen in our region, causes a lot of illness in children. And the related parasite, Plasmodium knowlesi, is actually a zoonotic pathogen from macaque monkeys, which is of increasing significance in Southeast Asia. Of major concern for the world today is that our efforts to control malaria have started to stall. So at the turn of the century, we were having around about 1 million malaria-related deaths per year. And through the widespread introduction of mosquito bed nets and better drugs, that halved in 2011 to around about 500,000 deaths per year. We really haven't been able to push any significantly lower since then. Of major concern is the development of multi-drug resistant malaria parasites, which are spreading from Southeast Asia into Africa. We're seeing increased evidence of climate change, changing malaria virulence in certain countries, political instabilities, and who knows what COVID-19 is going to cause to malaria control measures going forward. So it's certainly not something that we've solved to date. Malaria has a really complex life cycle involving human liver cells, mosquitoes, at different stages of development. But for the purposes of today's talk, I'm really going to focus on the blood stage of the parasite's life cycle, and that's because that's the stage that causes all disease symptoms, and also the stage we work on in the lab. So to start off the blood stage of malaria, the malaria merozoite comes along and binds to the outside of the red cell. It then pulls the red cell membrane around itself as it enters, forming a vacuole. Within this vacuole, the parasite begins to grow, begins to fill in the entire red cell, and then begins to replicate, forming somewhere between 16 to 32 new merozoites. These merozoites then burst out of the red cell and go on to invade a new host red cell every 24 to 48 hours. I'll just point out this black crystal. The significance of that crystal is that the parasite feeds on the host cell hemoglobin. When it does so, it releases free iron, and to protect itself from the free iron, it has to hide that free iron away in the crystalline form called hemozoin. So our lab covers uses gene editing to look at new vaccine approaches and also to uncover new biology, but a large part of our lab looks at new ways to develop anti-malarial drugs. Recently, in collaboration with Christian Doeg's group at RMIT in Melbourne, we identified that malaria parasites need host red blood cell kinases to survive. Now, kinases have essential and dynamic roles in signalling, not least of which in host erythrocytes. Kinases transfer a phosphate from ATP to a protein which can activate or potentially deactivate the protein. Human kinases are major associated with a number of human diseases, including cancers and also neurodevelopmental diseases. And they're of increasing importance for drug development, with over 60 FDA-approved human kinase inhibitors available today. Using a phosphoarray that measures the amount of phosphorylation of proteins, human proteins, we showed that the malaria parasite infection will upregulate and downregulate phosphorylation of an entire range of human proteins. A lot of these proteins are kinases, and kinases often act as a signaling hub and require phosphorylation for their own activation. We also found that the specific kinases activated differed between different stages of the parasite life cycle. So in early development, they differed to the middle stage of development. This stage is a stage of rapid feeding and expansion, and we found the most activation here, but also a different set of proteins were activated at the late stage of parasite development, immediately before they ruptured. So in all, we found around about 150 human kinases that are activated or deactivated with malaria parasite infection. We also found that host-directed kinase inhibitors, such as this MET inhibitor or this MET-ALK or these RAF inhibitors, were relatively potent first-pass drugs for killing malaria parasites in vitro. When we tested one of the MET inhibitors in an in vivo model of malaria, we found that the drug could control parasitemia. These are fairly old drugs and they're not best in class by any means, but they're proof of principle that we can use human targeting kinase inhibitors to control malaria parasites. So why would we want to do this? Well, one reason is that if we target a human protein, then it reduces the chances the parasite can have a mutation that leads to resistance. Another is that the human kinase development pipeline with the pharmaceutical companies is really a lot of investment has gone into there. Potentially then we could develop drugs cheaper for malaria. What we're working on now is the problem of different malaria parasites infect different age red cells. So Plasmodium falciparum can infect red blood cells of any age, whereas Plasmodium vivax can only infect very young red blood cells called reticulocytes. Now reticulocytes are a lot more complex. They have a lot more systems that are more similar to nucleated mammalian cells. And we find that there is a change or differences in the expression level, the phosphorylation of different kinases between reticulocytes and omocytes. We also find that there's a difference in phosphorylation patterns between Plasmodium falciparum malaria and Plasmodium knowlesi, which is another reticulocyte trophic malaria. So what we're embarking on now is trying to identify kinase pathways that are required for parasite growth for both reticulocyte trophic and parasites that can infect red cells of any age, so that we can make pan-malaria drug development strategies. Moving to some of the basic biology work that we're doing in the lab, I'll just introduce a few parts of the malaria merozoite. So there's a range of merozoite surface proteins, and then there's these two sets of organelles, the micronemes and the rhopteres. These organelles secrete proteins during the process of invasion that allow the parasite to bind to the red cell and also allow the formation of the parasitophilus vacuole that the parasite then lives inside. So as represented here, as the parasite binds, it releases proteins from the rhoptere and microneme organelles. The invasion motor pulls the parasite inside, the red cell fuses behind, and these proteins from the micronemes and rhopteres go towards forming the parasitophilus vacuole. All this happens within about 10 seconds. It has to be tightly coordinated. If something goes wrong with any of these complex systems, then the parasite can't invade and it can't cause disease. So we became interested in one of the proteins that we think has a role in rhoptere secretion. To investigate this, we used gene-edited parasite lines that attached a HA tag to the C-terminus of this protein, cytosolically exposed rhoptere leaflet interacting protein 1 or SERLI1. This allowed us to examine localization of this protein by immunofluorescence assays and also to examine protein expression levels by things like Western blot. Also attached to this gene editing construct is a glucosamine-inducible GLMS ribozyme. When we add the sugar glucosamine to our parasite growth media, it binds to the GLMS ribozyme of the mRNA for the protein. It causes self-cleavage of the GLMS ribozyme, removing the poly-A tail. And when you remove poly-A tail of the protein, it marks that, of the mRNA, it marks that mRNA for degradation. So if the mRNA is degraded, then we lose protein expression. So this was quite an effective method to knock down protein expression. When we knocked down protein expression, we saw a reduction in growth. And when we looked at it a bit more closely, we could see that it was actually the growth inhibition was caused because the merozoites could no longer invade the red cells. So we're interested to see whether the SERLI1 protein actually localized to, because that may give us an indication of function. The rhopteres actually have a bit of structure to them. They have a neck, and this neck region mainly contains proteins involved in initial binding to the red cell, and a bulb region which mainly contains proteins involved in forming the parasitophagous vacuole. Using immunofluorescence microscopy and comparing localization of our protein SERLI1 to the bulb marker RAP1, we could clearly see very close co-localization between RAP1 and SERLI1, indicating that our protein SERLI1 lies in the rupture bulb. The question then was, well, was it on the inside of the rupture bulb? If it's on the inside of the rupture bulb, it could be secreted and could be a vaccine candidate. Or is it on the outside of the rupture bulb, in which case we're probably better off, if we're looking at a therapeutic angle, to target it with drugs. So again, using super resolution fluorescence microscopy, we measured across the diameter of the SERLI1 and RAP1 co-localization, and consistently saw that SERLI1, the diameter between of the SERLI1 donuts was much greater than the diameter of the RAP1 donuts. And this, along with a number of layers of biochemical evidence, strongly suggests that SERLI1 is on the outside of the rupture bulb. So if we're going to target this with therapeutics, we have to think in terms of drugs. So why would we bother thinking of targeting the SERLIs with drugs? Well, it turns out the SERLIs are really conserved amongst malaria parasites, so there doesn't seem to be a lot of opportunity for the protein to mutate. And they are really unique. They're only shared between malaria and its closest apicomplexan parasite relatives. And so our work going forward will look to investigate whether we can target these proteins and their interacting partners with drugs to block host cell invasion. I'd like to thank my amazing lab team who have done all this amazing work and a whole range of collaborators around the country and also overseas who have made major contributions to this work. Thank you.
Thanks so much, Danny, for an excellent presentation. Danny, just a very quick question in relation to the idea of targeting a human kinase to kill malaria. Can you give us a sense of what that would look like in terms of, given that malaria is largely a disease that affects Africa, in terms of rolling out a therapy that would be not only affordable but also would offer the greatest opportunity for compliance of taking that drug? Can you talk to me a bit about the thinking around how that would look from an application process?
Okay, so one of the advantages of this strategy potentially is we're talking about drugs that have already been made, have already been moved through phase 1 or 2 trials, and then perhaps they aren't as good for their application as something else that the pharma company wants to push forward. So we're looking at orphan drugs, drugs that may not have a use going forward. We're also looking at a treatment regime which is only perhaps 2 or 3 days. So as opposed to using cancer chemotherapy, for example, where you could spend months on it, over time you are more likely to see side effects. We would be combining this drug with another fast-acting anti-malarial to knock the parasitemia down flat, kill the parasites, and then the drug is allowed to be cleared after a fairly short time. Potentially this offers major advantages in terms of not having to find a new chemical entity to try to from scratch to try to develop, and also to have a lot of information available already for in terms of their use clinically and their safety.
Thanks, Danny. Look, in light of the time, I just wanted to close up by thanking our 2 speakers today, Professor Michael Beard and Dr. Danny Wilson, for excellent presentations. And before we close, I just want to remind people that this mini review was recorded and is available through the Adelaide Biomed City website. And all previous mini reviews are also available at www.adelaidebiomed.com. And you can find the webinars under a banner suitably titled Webinar. So thank you again to our speakers today. Thank you for joining us online. And please join us next week at the same time and same bat channel. Thanks ever so much, everyone. Cheers.